Hello students, welcome you all to a new video of Sunanda's tutorial. So in this video we will discuss about protein targeting and sorting. So this phenomenon is otherwise known as intracellular trafficking. So do you know what is intracellular? That means any kind of transport inside a cell. It is the delivery of proteins to its final destination after translation. So how proteins are manufactured? Proteins are manufactured through transcription and translation. Transcription means the formation of messenger RNA from DNA inside the nucleus. It is the first step. Second step is the translation, where the mRNA gets decoded in using ribosome to protein, to a linear chain of proteins. Proteins are the linear chain or the polymer of amino acids. Okay. So after its formation, through protein targeting and sorting, the proteins get delivered to its final destination. This process can be achieved through two methods, either co-translational and second one is post-translational. Co-translational, that means during translation. Second one is post-translational, that means after translation. So we will discuss these two methods one by one. So at first, what is protein targeting? Protein targeting and sorting is a biological mechanism by which cells distribute newly synthesized proteins to their correct destinations, such as organelles, or it can be membranes or extracellular space. Driven by internal signal sequence, this process uses co-translational during synthesis and post-translational after synthesis pathways to ensure proper cellular organization. So here this process uses a signal sequence which direct it in a particular direction. Key aspects. What are the main important things to remember? Targeting signals. So proteins contain specific targeting sequence, otherwise known as signal peptides, which acts as zip code. Like we have our pin code, suppose 75100, something like that. So pin code means that particular letter to be delivered to that particular address or pin code. So pin code is the specification of that address, particular address. So that zip codes recognized by receptor that guide them to their destination. So what is signal sequences? These are short stretches of amino acids that detect the final destination. What is proteins? Proteins are chain of amino acids. So it is the primary structure of protein, linear protein. Primary structure of protein is a linear chain of amino acids. So this linear chain have a short stretch of amino acid. So there are two important pathways of protein sorting and targeting. One is co-translational, which is the second method of protein formation. Proteins get manufactured in two steps. One is transcription, second one is translation. During translation, proteins are targeted to their destination. Proteins destined for endoplasmic reticulum, Golgi complex, lysosome or secretion are synthesized on membrane bound ribosome, where they enter the endoplasmic reticulum during translation. So SRP, signal recognition particle, it recognizes the signal sequence, halts translation and docks the ribosome to rough endoplasmic membrane. Then the particular proteins get transported or transferred to their particular destination, like Golgi complex or lysosome to any secretory pathway. Second important method is post-translational input. Proteins for cytoplasm, nucleus, mitochondria or peroxisome are synthesized on free ribosome and imported directly to organelle after synthesis is completed. So this is the post-translational. Okay. So one method is co-translational targeting, which is otherwise known as CTT. Co-translational targeting or secretory pathway. Another one is PTT, post-translational targeting. Okay. It is the non-secretory pathway. Takes place in nucleus, mitochondria, chloroplast and peroxisome. Okay. So what we have learnt that there are two methods. One is PTT, that means post-translational targeting, in which the steps are binding of cytosolic chaperones. So chaperones are there to protect proteins, then recognition by receptor proteins on destination organelle, then translocation or transfer through membrane channels, then finally cleavage of targeting signals. So all this in post-translational targeting takes place after translation. Then CTT means co-translational targeting. That means when translation continues, after formation of one protein, translation halts for some times and translation stop to start targeting. After targeting of that particular protein is completed, then again the translation restarts. So what are the steps? Recognition of nascent signal peptide by signal recognition particle, halting synthesis followed by docking at the membrane, and translocation through translocon channel in case of co-translational targeting. So there are steps like translation start, then translation halts for some times to start targeting. So these two diagrams are the diagrammatic representation of protein targeting and sorting. So this is all about protein targeting. I hope you will get benefit from this video. If you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial to get more important videos like this. Thank you.